And may I introduce you now to Professor Frances Platt. She's a professor of biochemistry and pharmacology and also heads the Department of Pharmacology. And today she will talk to us about unanticipated links between rare and common neurodegenerative diseases. So really quite an interesting topic. Welcome.
Thank you very much. Can you hear me? So thank you for the kind invitation, and this is a very different topic from the previous excellent talk, so a change of gears here. So we work on rare neurodegenerative diseases, and over the last few years it's become clear that there are some mechanistic links to particularly Parkinson's disease. And so for the sake of time today, I'm going to focus on that story. So the diseases that we work on involve some aversive stimuli, which are sphingolipids. And I think when most people see these kind of schematics, and it's sort of off-putting, but essentially sphingolipids are a complex group of lipids that exist in the cells. They've evolved early in eukaryotic evolution, and they have this ceramide backbone that's comprised of a sphingosine sphingoid base and a variable chain length fatty acid. And so this on here is just a schematic of this. If I can get this to work. This is ceramide, and it can be modified by the addition of different head groups. And the family of molecules we study are glycosphingolipids, where there's a carbohydrate head group. Now, there are a subfamily of glycosphingolipids that are called gangliosides, and these carry charged sialic acid residues, and this will become relevant later on. Now, there's nothing simple about this family of molecules, and their cell biology is also complex, but let me just break it down into some simple components. So the ceramide moiety is formed in the endoplasmic reticulum. It's transferred either through a non-vesicular or vesicular route to the Golgi apparatus, and then sequentially you build up the sugars to generate glycosphingolipids. And these lipids, we still don't understand their functions, but they're believed to primarily function at the plasma membrane. And in the brain, there are these 4 dominant gangliosides shown in this box, and the functions of these are debated, but they are important for neuronal function. When the plasma membrane periodically is internalized for a catabolic turnover, you get the endocytosis of these glycosphingolipids. They go through the endocytic system, and they end up in the lysosome, where they're sequentially degraded. So it's sequential biosynthesis, sequential degradation. Now, this is the enormity of what we're dealing with. This is a lipid maps schematic showing all of the different structures, and there's about 300 or 400 structures that have been described. These don't all occur in mammalian systems, but it just shows you the combinatorial diversity of these carbohydrate head groups. Now, for many years it was thought that there were no biosynthetic disorders, because if you knocked out the first gene in glycosphingolipid biosynthesis, it was embryonically lethal. But the question was, were there human diseases where it might be a downstream defect to do with head group formation? And it was discovered through the work of Andrew Crosby in collaboration with David Priestman in my lab, that in an old Amish population, he identified the first biosynthetic disorder in this family, which is a GM3 synthase deficiency. GM3 is one of those charged gangliosides. And these patients have a very, very severe epilepsy. And the existence of these patients suggested that the imbalances of these glycosphingolipids, particularly these charged glycolipids, or the charge to neutral ratio of these glycolipids, was important for neuronal stability and function. A few years later, he mapped a second gene in this pathway, which is called GM2 synthase, and this actually presents as a spastic paraplegia. So 2 very similar diseases, and in these boxes, it just shows you the different glycolipids that are lost in these 2 disorders. So you can have biosynthetic diseases, and I just want you to remember GM3 synthase, because this is going to be relevant later in the talk. In contrast to these two biosympathetic disorders, there is a fairly large number of catabolic diseases that have been known for many years, and these are called lysosomal storage disorders. And this is what we study primarily in my lab. And this is a sequential degradation of glycosphingolipids. And if we just look at the last step in this pathway, there is a disease for most of these steps in the pathway. These are largely enzyme deficiencies. And Gaucher disease is the last step in the degradation of complex glycosphingolipids. And it's the removal of glucose from ceramide. And this enzyme is particularly interesting because it has dual activity. It's a hydrolase, it removes glucose from ceramide, and that's what most people associate it with. But when the enzyme removes the glucose, it transfers it to another acceptor. So it's a transglucosylase as well as a degradative hydrolase. And it puts that glucose onto things like cholesterol and eicosanoids. So in Gaucher disease, the enzyme deficiency means that you build up glucosylceramide, which is the substrate for this disorder, but you will also have a deficiency in these glycolytes, these glucosylated metabolites that this enzyme also makes. So if you look at lysosomal storage diseases as a family, they are inherited as autosomal recessive traits, so you need to have both alleles mutated. There's about 70 of these disorders that have been described, but that number is growing. They affect about 1 in 5,000 live births at a collective frequency. And the sphingolipid storage diseases are one subfamily, which is the family that we study. The other members of this family involve the different biochemical substrates building up in the lysosome. The majority, but not all of them, are defects in lysosomal enzymes. There are also membrane proteins in the lysosome that can be deficient and cause these disorders. Most of them have CNS pathology and neuroinflammation, and some of them have approved therapies, which has been the basis for a lot of research in my lab over the last few years. Because you have lysosomes in virtually every cell of the body, these are multimorbidity diseases. These are the different genes in these different boxes, and just to notice that the brain is the largest box. So if you don't have an efficient catabolic system in the lysosome, you're prone to neurodegenerative diseases. But if you're thinking about treatment, you can't just treat the brain because there's also peripheral organ involvement. So that's why we've been focusing on small molecule treatments for a number of years in my lab. So what's the link between these disorders and Parkinson's? So the start of this story is interesting because it didn't come from GWAS studies or anything of that nature, because these are rare alleles in the general population. It was actually from a clinical observation. And that was that Gaucher disease patients who have an effective enzyme replacement therapy go for infusions periodically, and they are very well known by the clinicians who treat them. And the clinicians, the specialist clinicians who see these patients, notice that there was Parkinsonism in the patients themselves, but more interestingly, the family members, the carriers of these mutations, also presented with Parkinson's. And for many years, this was hotly debated as to whether or not this was just chance or whether there was some mechanistic link. The frequency of Parkinsonism was found to be higher in GBA heterozygous carriers, and that was proven, and GWAS studies confirmed this. And if you go and look for, in general Parkinson's disease populations worldwide, you find that mutant GBA alleles are one of the highest genetic risk factors for developing Parkinson's. So in homozygous state of mutation, this causes Gaucher, and heterozygous mutations predicate for Parkinson's disease. And the big question mechanistically was, is this link something very specific to do with GBA mutations and Parkinson's? Or is this the canary in the coal mine, because Gaucher is one of the commonest of the lysosomal diseases, and so the carrier frequency in the population is low, but it's higher than a lot of other rare lysosomal diseases. And so Josh Schulman at Baylor did the experiment the other way round. He collected a cohort of about 2,000 sporadic Parkinson's patients, and he went and sequenced them and looked for the 60 genes that cause lysosomal storage diseases, and he looked for known disease-causing mutations. So what he found was that over half of those 2,000 patients had one or more heterozygous mutation of a known disease-causing allele associated with one of those 70 lysosomal storage diseases. And several of his patients had 1, 2, 3, or 4 heterozygous mutations in different lysosomal disease genes. So what Josh had suggested in this study was that it was lysosomal dysfunction that was the risk factor for Parkinson's. And the question then is, why do heterozygous mutations, why are they the risk factors? Why is the dopaminergic system and other circuitry affected in Parkinson's so vulnerable to these heterozygous mutations? Was it gain of function or loss of function? So we had a look a few years ago at the substantia nigra from post-mortem brain samples, both control and Parkinson's. And what we found was that there was an elevation in glucosylceramide. This is the lipid stored in Gaucher disease. So this was consistent with a loss of function mechanism. But to our surprise, we found that there were several other changes in glycosphingolipids, and most of these was actually a loss of normal levels of these complex charged gangliosides. So we had reduced, we had multiple perturbations in the pathway, not just at the level of glucosylceramide. And when we looked at the lysosomal enzymes in these Parkinson's patients, what we found was that the GBA activity, this is the Gaucher enzyme, was indeed significantly reduced, but there were multiple other lysosomal disease-causing enzymes that were also reduced. So there's multiple changes going on in sphingolipid metabolism in these Parkinson's patients. More recently, in collaboration with Per Svenningsson at the Karolinska, we looked at a large Swedish cohort of patients, and we found the same changes that we'd described previously. But in this study, we were able to stratify the patients based on whether or not they had, so these are the controls, these are low glucosylceramide storing individuals. This is now looking in the plasma, and this is the high glucosylceramide stratified patients. And when you look at the changes, for example, if you look at GM2 ganglioside down here, it's down in both populations. And this was true for other glycolipids as well. So it's not that the change in glucosylceramide drives secondary changes in other glycosphingolipids. These things seem to be independent of one another. Now, one of the things that caught my eye in Josh Schulman's original paper was that Josh had done this experiment where they had gone and looked for lysosomal disease genes. So they had classified all of those genes and then looked for mutations in those genes. And one of the genes that they had looked for was a thing that they called GM3, is a gene that they said causes GM3 gangliosidosis, because the nomenclature implicates GM3 in the action of this gene. Now, GM3 gangliosidosis does not exist as a lysosomal storage disease. And what this gene is that Josh looked for is actually a bisynthetic enzyme. It encodes a bisynthetic glycolipid enzyme, which is GM3 synthase. And that's the same gene that causes this very severe epilepsy in the old Amish community that Andrew Crosby studies. So what this says is that this isn't just about lysosomal dysfunction, this is about sphingolipid imbalances. Because this is not a catabolic gene, the enzyme is working in the Golgi apparatus. And this implicates the fact that a common feature of Parkinson's is actually imbalances in glycosphingolipids. And I think the misclassification of this gene has actually fortuitously given us a real insight into the fact that this ultra-rare disease in the heterozygous state is also overrepresented in Parkinson's patients. So in summary, there are altered patterns of glycolipid expression in Parkinson's. And interestingly, these are just a more extreme version of what we see in normal aging in both rodents and in human. So we all have age-related declines in lysosomal function. We have age-related declines in sphingolipid degradation. And so levels of glycosphingolipids change as we age. And what's happening in Parkinson's is that that's just more extreme. So the loss of these enzyme activities is greater. And so we think that these changes in lipids that you can pick up in any tissue of the body, you can pick them up in CSF, you can pick them up in plasma, you can pick them up in post-mortem tissues, because these are lysosomal changes, and lysosomes are in pretty much all cells of the body, you can potentially use these as biomarkers for stratifying patients and potentially for looking at relative risk. The changes in complex gangliosides that we think may be risk factors for Parkinson's, or are risk factors for Parkinson's, are independent of glucosylceramide. So I think that this is not just about GBA and glucosylceramide. There's a much, much more complicated picture going on here. So what we're doing at the moment is that we, through a Michael J. Fox Foundation grant, an ASAP grant with John Hardy, we're looking at these sphingolipid changes as possible biomarkers in both plasma and CSF in sporadic Parkinson's cohorts through the Fox Foundation. With Hugh Morris and John Hardy at UCL, we're looking at how these lipids change and do they correlate with progression, and we've got some interesting preliminary data that says that they do. And then we're also trying to look at whether or not there are novel targets for therapeutic intervention based on sphingolipid imbalances, and we've been doing that in collaboration with Pfizer. So I will finish just by thanking the team in the lab, particularly Kerry Wallon, Danielle, Mylene and David, who have done most of the Parkinson's work, and our collaborators both at the Karolinska, Harvard and also at UCL. And I'm very happy to answer any questions. Thank you.
Thank you very much.